open the curve note. So I hope you already finished this chapter 7. So this is about data parsing. We need to test all the code and add more information if we need image like this. Make sure that the image is also well sized, not too big like this. Like this is too big, yeah. So good, this is already tested. If we look on the whole plan, uh, we are here. So we need to test all chapter before to make it more consistent. I checked this morning, like this is too short, this caption. Need to add more caption. Usually the best way to make this caption is just to ask GPT, capture the image and make caption in one sentence. So it's not too short like this, okay? The other thing is the picture. Make sure that the size and aesthetic is good enough for the chapter. Okay, I hope we can finish up to chapter 12 as a part 1 of this book. Part 4 will cover more NF4 ready pipeline in Nextflow, that we can change some code inside existing pipeline to make it faster. So maybe we put this part as a part 2 of the book, and let's release this part 1 of the book up to chapter 12. Okay. So today we will enter chapter 8. I already have a draft, and it's okay to start the image and also test the code. But in this chapter is starting to get more advanced because we are entering AI-driven analysis. Means that some code maybe have AI code based on Rust. Okay. So in this chapter, we cover variant analysis and annotation. So variant analysis is like a computational workflow to detect, annotate, filters, and interpret genomic difference compared to some reference. It can be single nucleotide variants or indels or structural variants and others. So it starts from raw sequence read and pipeline alignment, and then call candidate variant with statistical and machine learning, and then assign quality metrics and make sure that the result is good enough statistically. And in modern way, we sometimes use machine learning and deep learning in the pipeline. Okay. So modern bioinformatics is no longer only matching against single reference genome, but it's also reason over graph like pangenomes, pangenomes, and some pipeline have deep learning caller. Okay, it run in CPU and GPU cluster because deep learning is based on neural network, so it should have GPU to make it faster. So in this chapter, we start with explaining the classical Hardy-Weinberg framework. So let the P as a reference allele frequency and Q equal to 1 minus P. Okay. So under the, we need to define the probability of PRAA P quadrat and PAA 2PQ and then get this formula. For n individual, we expect the counts to be like this. So The genome graph is a specialized graph like a site, which is bubble alternative path from past FASTA alignment. So it's a very common method to use Hardy-Weinberg equilibrium techniques to quality control. So this is explain the background. So there's many variant class like SNP and short indels, structural variants, mobile element variant, and selection scan. That's why if you go to Nextflow, there's a lot of types of variant calling here. Let me take, for example, the deep variant. This uh, is already archived, no longer maintained yet, this one. So this is analysis pipeline to detect the germline or somatic variant. So now variant calling is becoming very much complex because of the additional statistical method and machine learning method. If you see here, it's a very good picture to describe the pipeline process. So from the pre-processing side, we need to convert the data source. Let's say it's not FASTQ, then need to convert to FASTQ and then perform UMI, FASTQP mapping, and then check on BAM and PRAM, and then check duplicate, uh, mark the duplicate. So this is kind of a pre-processing to make sure that the data coming into the variant calling element is in good quality. So once you put the variant call the variant call, there's also pipeline here like variant calling algorithm, BCF tools, VCF tools for checking, and germline, and then output the VCF. After that, there's annotation again to make sure that we have the final VCF. Okay, this is just one example. There's many more in NF Core as a repository of the pipeline of Nextflow. Let's check another example. So this is GTK base for RNA variant call. Okay. So it's a pipeline for RNA variant calling analysis based on GATK4 best practices. If we summarize like this, first is merge sequence FASTQ file, read QC, extract UMI, align reads, right? So each variant usually have some pre-processing and variant calling element in the pipeline. And we can reuse instead of build the pipeline from beginning. Our plan is actually not to rewrite everything in RAS, but describing how RAS can implement some part of the pipeline, like some process in the pipeline, to make it faster, so not to replace. So in this case, for example, let's say if there is a method that we can make it faster using RAS, then that part we can implement in RAS. Okay, why that is possible? So let me show. Let's say we try this select, okay, and go to this pipeline, and then let me create new project in my here. And I say analyze the following Nextflow pipeline and give report on key process, key task in pipeline that can be accelerated by using Rust instead of Python. Okay. So I asked the Clouder to analyze this GitHub and come up to me uh, what kind of component or task that we can accelerate if we convert to Rust. Okay. So AI like Clouder now can go into this and then scan the data, scan the code, reading the code inside, and then let us know which area that maybe we can improve if we port that code into Rust. Of course, it will take some time for it to go to the GitHub, but we hope that in part 2 of the books, we can perform some optimization on that card. It cannot access the GitHub. Wait. But this GitHub is open, right? Let me check. This is open GitHub. Oh. Okay. So let's see. So it cannot access through the API, but it can show you pipeline overview. Okay. And show the file and indicate this is high CPU, high memory. Okay. So indicate this moderate CPU, but can be bottleneck. This optimization is also required. So this is the important result. So it can tell you which task in current Python environment or baseline code and how many percent Rust improvement we can expect from this. If we turn the Python, let's say, VCF annotated all, yeah. Let's say we take this, take the code and re-implement using Rust code and best related trade. That will go to the artifact of the Python code and then take the Python code and then re-implement in Rust. As you already learned how to mix Rust into the Nextflow code, then you can take this code and then re-implement the pipeline using some performance improvement using Rust. So this is what we expect in part 2 of the book. Okay, that's why in this chapter we need to provide more comprehensive sample code to make sure that we can improve the performance of the Python code. So this is example of the Rust implementation of genetic data analysis for variant called VCF file to calculate the Hardy-Weinberg equilibrium statistic. So as you see, it used Rayon. This is a parallel CPU. It used Polar, is a high performance zero copy CPU GPU. And this code is quite long, but I hope you can test and improve. This code is written by Clouder, so it's recommended if you bug fix using Clouder instead of using ChatGPT. Okay, so this is just an example. and then it gives you the next flow run code. Let's say for Sarek, yeah. Maybe we have to change this because it's not really running the Sarek, but is just a code snip. Yeah, it's not full pipeline running, but it's a good overview of how we can improve the next flow pipeline in the future using Rust as a language for task implementation. Okay, so in section 8.2, and then we cover the data structure for variant representation. So storing the variant information into a data structure that is efficient and can be computed efficiently is very important to learn. Okay. So usually we use the Burrow-Wheeler transform or graph of GBWT is a structure that we use to store the information inside the memory. Okay. I think there's some information about this graph in the previous chapter, so it's not elaborated here. So in this subsection, we focus more on how to find the modern pipelines that are utilizing modern data structure. Okay. So for example, if we do variant calling format, how to store the data of VCF inside the structure in memory. Okay. So the code example here is we want to perform pan-genome analytics using Rust. Maybe we need to remove this one. Yeah. This is the Rust version. So expanded the capability of the CPU. This is the SIMD capability of the CPU. So instead of using the traditional VCF processing, okay, this code will employ the GBWT, yeah, and for the representation in the memory, and then provide the high performance version of the storing the data into the memory. Again, the code is quite long, 433 pages. Yeah. So during reading this section, you need to make sure that if there's any typo or mathematics that is not put into LaTeX, please help to fix. Yeah. But this is kind of how we store the VCF data into high performance structure using Rust. Okay. And section 8.3 covered the algorithm for variant call. So in many variant call, this is the most important after pre-processing that we need to have the algorithm for variant calling. So it's statistical modeling, but using Rust, we expect it to be more high efficient. And of course, because machine learning is a part of the statistical model, maybe we already have many variant calling is contain deep learning method or deep learning model. So let me check. Maybe this is including a deep variant. This is, of course, this will have the variant code. Let me check. Okay. Okay, this is already Word. Analyze this NF pipeline and explain the deep learning method used in the variant calling algorithm. So instead of we go to the code manually, I think AI now is doing that better for us. I hope it can go to the GitHub folder and find out the algorithm that it used to implement the deep learning method. Okay, let's wait. Okay, it takes some time. Okay. So this is the mathematical background of the bias theorem. This is come from probability. So the likelihood of PD given G, it calculated like this. Okay. Check the mathematics, improve if you can. And in this table, is explain the common method that people use for this task, for algorithm for variant calling. And if you see here, there is a pile up sums tools. The core idea, like count-based proposition using binomial and beta-nomial tests. It's quite fast and simple, but poor in terms of indels. Holotype assembly, haplotype assembly is coming from GTK. Is the core idea is use the De Bruijn graph. Okay, it's quite accurate for SNP and indel, but limitation is CPU intensive. Another method is graph genome, like VG and Scalo. Is a variation of graph encode all parts. Okay, overhead is graph construction, and deep variant of like what we show here in this GitHub. Okay. So different convert three pipe apps to haplotype image into tensor and perform CNN or transformer to predict the genotypes. And Illumina PacBio use this strategy. Of course, it will require GPU. So if you design a machine for this kind of computation and you put GPU inside, maybe you can accelerate the CNN or transformer code inside if you use RAS. Okay. So in this code, this is just example. So this is RAS-based genomic variant caller. Okay. I start from BAM or CRAM file, implement fast parallel processing from BAM or FASTA. It use polars, and this is the code. Quite long. 750 line of code. Okay, it's quite long, but what to try to learn how they can implement, how it can improve performance, for example. Okay, so let us check our repo here. So using tools, AI tools that can dig into the GitHub, you can check the pipeline in NF Core and ask the AI to find out which area in the pipeline, which task that we can improve using RAS. Okay, so this is the result. One, deep variant. Yeah, so it is a DNA sequencing transformation. Use CNN. Yeah, and to classify the genomic position as reference. Okay, and also heterozygous variant and homozygous variant. It's used convolutional neural network. Just in case you can understand the convolutional model and see the implementation, like this is 1D CNN and 2D CNN. If we can get the code and ask it to port the code, yeah, to RAS, we can change inside the implementation. Okay, let me check. Can you re-implement the models from Python to Rust to gain performance. You can go again to that folder and implement using Rust. As you see here, it used PyTorch, Rust version of PyTorch. Okay, that goes there and implement. And if you see the implementation, you have opportunity to control the type of variables, make sure that you gain performance, you can perform quantization, you can perform additional optimization techniques to make sure that your 1D and 2D CNN models can perform much faster in your machine. Okay. So in this code, yeah, just give some illustration on how we can improve the performance. Next is variant annotation principle. It's the algorithm, again, statistical model, to translate the raw variant call into biological meaningful statement, like, for example, which gene or regulatory element is affected. So this kind of like annotation principle. Or other question like, does the change disturb protein structure or RNA splicing or transcription factor binding or population health? Okay. This also contain many AI pipeline in the future. Okay. So for example, let's give you example here. Okay, we also will have problem later on with some citation, but don't worry about citation for now. If you find this kind of citation, that means I already put the reference in this reference side. Okay. If I keep the citation in the section, I already have it there. So just keep like that in the text. Okay, let me go section 8.4. Okay, this explain the basic mathematics, explain the state-of-the-art tools like this one, yep, and give code example of implementation for genomic variant annotation pipeline from multiple data source to predict the pathogenicity of genetic variants. Okay, this code also quite long, 800 code, but you can just copy paste and then try to run it as long as it has the main method and you can supply the data and then you can try to make it run. Okay. So the next chapter, the next section is just how to integrate variant analysis into Nextflow pipeline. As you can see in NF, many pipeline already there. So what we need to understand is what task inside the pipeline that we need to improve using Rust. Otherwise, you just run the Python code without any performance improvement. Okay. So if you get the source code of the pipeline in the GitHub, you can ask AI to find out which area in the code that you can improve, and then you can improve the code by migrating the code from Python to Rust. Okay, so this is the example for 1D, CNN 1D, and CNN 2D implementation. Okay, so it tells you where to put in the Nextflow code. Okay, so I think it's possible in part two of our book, we improve the pipeline. But in part one of the book, we just explain the specific task as an illustration. Okay. Okay. In this section, 8.5, we also explain, give some sample code for complex workflow processing. Yeah. For generating sequence data through alignment, variant calling, and annotation. So three process inside one code, but is a modular uh, command line program that can take uh, nodals BAM and nodals VCF file inside the pipeline. Also very long codes. Hopefully it works. Okay. And the last one is just an advanced topic, illustration of advanced topic in variant analysis. It gives you example of AI-driven pipeline. It's quite advanced because it's already used Onyx. So Onyx is kind of machine learning inference. So you already have the machine learning and you infer the machine learning. So it's quite advanced example of how you use machine learning inside the variant calling pipeline. And the code is also longer, 967 line of code. Okay. So that's all for chapter 8. It's quite long in terms of code, but as this book will be published as a web books, not printed books, don't worry about the length of the code because people can just copy the code and pass to their IDE. But make sure that the code can be tested well. And if you think the explanation is not precise or too dense or too short, you can also expand the explanation. Okay. So that's all for chapter 8. I hope you can perform this task. Proofread. Okay. You can also try if you have clouded. Unfortunately, I never try using GPT, but let's try. Let's try now. Yeah. So we can check. I'm not sure if GPT-3 can go into, but let's try. I put search to GPT-3. Can it go to search the GitHub? Looks like it can. Assuming it can browse the file, so it can find the Python implementation inside the GitHub and analyze it. You have to use paid version of GPT to get the features, the GPT-03, but this is the new version of GPT-01 Pro. So 01 Pro becoming legacy. So GPT-3 is better in advanced reasoning. You can also try with GPT 4.1, for example. Just in case you don't have the O3. Wow, the weather in Depok is really bad. Storm. Okay, still working. Okay, it's now done. So now give you the report. So just like in cloud, it gives you the tasks inside the pipeline that worth to rewrite in Rust. So let's say variant calling. Why it's slow? Because it's can BAM, yeah, using Python loops and NumPy Blast. If you change using Rust, you can utilize Nodal and HTS Sleep, high throughput sequencing library. Make sure that we can speed up 60% with less memory. Okay. Report. If you use polars, you can expect this. Okay. So it's quite interesting. Yeah. So maybe you should try if you want to write papers for your master degree. You should try one common use pipeline and choose the task inside the pipeline and then change to Rust with best grades and compare the performance. So I firmly believe that this approach can improve performance without changing the whole pipeline in next flow pipeline, let's say Sarek or deep variant. Okay. We just pick specific tasks, porting the code to Rust, and make sure that the whole pipeline run again and expect performance gain. Okay. A lot of community around the world is innovating good pipeline, but as you know, creating pipeline and improving performance is two different tasks. Okay. While you are making the pipeline, you are not really thinking of performance. As long as it works, it just release. But it's really good opportunity if you build products and make sure that your products is having better performance compared to others. And many company is actually doing that, like 10 time genomics or PacBios or many other startups that actually looking for how to differentiate their pipeline, bioinformatic pipeline, compared to the open source that is available in NF Core. Okay, that's all for chapter 8. I hope you try. If you don't have the paid version, it's quite limited in Clouded because it limits your token per day. But if you have the paid version like this, I believe Fadin has. So as long as you know what pipeline you want to analyze, Fadin from Calbe Digital Lab have access to these tools. So you can utilize both this GPT-03 and Clouded. In term of code porting, I think Clouded is better than GPT-03, but need to compare the result because I think they also keep improving in GPT. Okay, so that's all for me for today. Is there any question regarding previous chapter and chapter 8? Let's discuss.
Hello, Mr. Risman.
Yeah.
I tried the same prompt in cloud and but outside the project, so general chat, and it can fetch the GitHub and analyze and
Mhm.
Python. Maybe I will share my screen.
Okay. So first prompt is to identify Python code that can improve, right? That can be ported and how much the performance gain you can get. The second prompt, given the Python code, translate to Rust with related crates, right? And then the last prompt, how to integrate back into the previous pipeline in Nextflow. Okay, show us, Fadil.
Yes, so the same prompt, but I using the general chat, just click new chat because I think with when we in the projects we can add project knowledge and connect to GitHub, and I think that's become the problem when the cloud went to retrieve the GitHub. I tried before, and it seems like when chat to in the general chat and want to port into the GitHub, it works better in the general chat. And in here, the the cloud can access searching the NF core Sarek pipeline. And in here, cloud also looking at the specific modules in each project structure like docs, usage, MD, main.nf, and others. And it also search several times like the specific information about Python modules that can benefit from Rust acceleration in here. And
Mhm.
here, uh,
Wow.
like Sarek did not perform variant calling, and here cloud retrieve all of the information related not only GitHub but searching on maybe some problem that people faces so it can improve with Rust. And in here, like they Rust in here, and it's many
Wow. It's
it's
very comprehensive.
yes, it's like multiple time. I think it's about 10 times like change log in here, and each result is keep logging on, and after that the analysis is being done, and based on the analysis the potential candidates first is interval processing and parallelizations to speed up pre-processing and also variant calling that you mentioned before, and for VCF file processing scripts, variant calling also, and input file parsing and validations. And at the end, implementation consideration. And the next prompt is to generate, I tried to prompting generate the optimized code using Rust for that GitHub Direct pipeline. And it kept uh, hit the max length for a message, but I just push the button continue like one, two, three, four time,
Yeah.
and the last time it will analyze the complete implementation for Rust optimization for NF Core Select, the summary and pattern implementation, the also the benchmark testing. User can expect the following performance improvement.
Wow.
Speed up eight, 18, 18 times and 15 times using Rust.
Wow. So I think it's a good opportunity for papers. So you pick one pipeline in NF Core that is commonly used and produce new version with Rust optimized code. Not all. We don't have to migrate everything, just with high potential speed up, right? And then release and then write papers. That's interesting. Wow.
Yeah. I'm shocked also that
Yeah, but we need to test the code, right?
yeah,
After you get the optimized pipeline, you can publish again in NF Core,
yeah,
and oh,
yeah.
that's good. Yeah.
Also give the project structure that we have to.
Wow. It's good time to become bioinformatician.
True. Yeah, I think that it's from me. Try on the Cloud 3.7 Sonnet using the
Maybe this is good for our final semester project. Let, let's say how many people in the team here? Let's say split into two pipeline and then people work to optimize and then you can make your draft papers, right? How to optimize a specific pipeline in bioinformatics and functional genomic using RAS. Very good demo, Hadin.
Thank you, Mr. Rishma.
But of course, need to be tested. Sometimes the code is hallucinated. So manual testing, final QC of the code before publishing again is required. But it's really good. I mean, Data Science Center can have this area, uh, improving performance of public pipeline in NF Core, which is globally feasible. The community is big compared to SnackMake or Galaxy or other community before. Okay, so please continue chapter 8. We will do the optimization in part 2 of the book, but maybe our final exam will about this. So work with Fadin regarding if you don't have access to Cloud or GPT. Cloud is better in terms of Rust code. I try many times. Cloud can do bug fixing faster than previous O1, but I never try O3. So it's good for you to have the vibe coding experience because code with AI will be the most wanted skill after you graduate. AI will be faster on writing codes, but without understanding of the data structure algorithm, you cannot instruct the AI to do what you want. Okay, before closing, any question, any feedback? No? Okay, please continue work on chapter 8. I will work on chapter 9. Let's meet again next week on chapter 9. Thank you, everyone.
Thank you, sir.
Thank you, Pa.
Thank you, Majere.